Hi, today in this module we will learn about automation in microbiology laboratory. A clinical microbiology laboratory are first line to combat and handle infectious disease and antimicrobial resistance, including the newly emerging ones. Although most clinical laboratories still rely on the conventional methods, a cascade of technological changes, if introduced, will revolutionize the management of clinical diagnostics. Let's see what is laboratory automation. Automation is technology by which a process or procedure is performed without human assistance. Therefore, in laboratory context, it means that every switch from manual work to machine can be called as an automation. However, in broad sense, all machine in laboratory are a kind of automation. A more acceptable example of automation will be a blood culture system which is continuously monitoring the growth of organism growing in it, or an automated MIC determining system will be a better example. Now, these are some of the individual automations. Apart from that, there are total lab automation which normally is used for the automation of diagnostic workflow, which includes all the essential steps including inoculation to the final results and reporting. Therefore, a laboratory which is performing complete automation has to process different specimens like containers, agar plate, broth and slides. At the same time, it has to inoculate various culture media and incubate. At the same time, they have to evaluate, document, and if needed, follow up the work if initiated. So these kind of fully automated laboratory are today only two companies provide a complete automation, and both the companies have or the systems have proven to show multifaceted benefit for the laboratory and the hospitals. Now let's see what can be the changes if these complete automated laboratory are introduced into the healthcare systems. So the changes are expected, like the demand of new tests will continue to grow, not in total number of tests, but also the types of the test which is going to be performed. At the same time, the balance between the molecular test as well as conventional test like conventional culture will definitely shift towards the molecular test. At the same time, current and future automation will be needed to incorporate both culture and molecular testing into the single specimen stream. At the same time, demand for more rapid turnaround time for infectious disease assay will also increase. So, clinical microbiology laboratory faces many challenges, and these challenges are also to be faced by the automated laboratories. So, let's see what are the challenges of automation. Clinical microbiology laboratory, since its inception, has been dependent on highly technical and skilled workforce to receive, process, and interpretation of the result. And this is because of the microbial processing of the system, which is a bit complex. This result in a poor turnaround time because the laboratory has to inoculate different media and await for the culture to grow. At the same time, there is increase in the number of specimens which are to be tested. Now let's see what will be the potential advantage of the introduction of this automation. There will be expected to increase the capacity of sample processing with better documentation and traceability. There will be better control of the cost both in terms of reagents and media with reduced turnaround time, and thus reducing uh, the turnaround time and giving a faster diagnosis. The automation is also expected to permit the extension of the opening hour of the laboratory. This will benefit hugely for the patient. At the same time, the laboratory will not be dependent on the skilled, trained professional, which is a loophole for any conventional laboratory. Now let's see about the technical innovation. The most influential technical innovation includes the introduction of liquid culture media in the clinical microbiology, which has an advantage that the sample is more homogenized in the liquid phase and it enables more consistent inoculation into the media. Now, other new techniques like liquid transport media, molecular techniques, and automated identification and susceptibility testing system have also simplified and allowed for greater standardization of the microbiology laboratory. Now, among the technical innovation that has driven the automation of clinical microbiology laboratory is the introduction of MALDI-TOF, that is matrix assisted laser desorption ionization time of flight mass spectrometry. MALDI-TOF procedures are highly amenable to automation because they are reliably simpler and do not change with organism and are reproducible. At the same time, they have revolutionized the microbial identification by providing a cost effective method that is also standardized. So this technique offers accurate, rapid, and inexpensive identification of microorganism which can be isolated from the clinical specimen. Now let's see the role of next generation sequencing technologies for automation. The NGS has represented a further milestone in the clinical microbiology laboratory. At least 4 main sequencing techniques are available, which are based on different chemistries for sequencing and that allow different outputs in terms of number and the length of sequencing rates. Now, let's see the importance of artificial intelligence. The amalgamation of artificial intelligence with next generation sequencing and automation has done wonders for the management of infectious disease. Computer visions with ability to process a digital image and identify object can be useful to improve the identification of the pathogen, removing manual tasks like interpretation of the gram stain. Now let's come to the conclusion. New technological advancement are going to change appearance of clinical microbiology routine laboratory with data increasing in volume and complexity. At the same time, implementation in real clinical setting should still improve an improvement in making process faster, cleaner than conventional workflow. With this, I come to an end of my talk on automation in microbiology laboratory.